Okay, welcome back to Chapter 12, Mendel's Experiments in Heredity. This is our second lecture in this two-part series. There are some overlapping of objectives here between Lecture 1 and Lecture 2, but we're going to learn about Mendel's principle of independent assortment. We're going to look at a dihybrid cross. So you know what a monohybrid cross is all about. Now we're going to look at a dihybrid cross and how to work out a dihybrid Punnett square. We're going to get the idea of test crossing, and we're going to differentiate dominance, incomplete dominance, and codominance. Okay, so we'll look at those in this lecture, in this lesson. So human pedigree. Some human traits and diseases are controlled by a single gene, and some of these exhibit dominant and recessive inheritance. So pedigree is a picture, a roadmap, if you will, that is used to track the presence or the flow of a particular trait throughout a family. So a dominant pedigree, all dominant traits appear in every generation. Recessive pedigree, recessive traits can skip a generation, but there are carriers, and we'll see that as we go through, especially with regard to, for instance, sickle cell disease. Okay, so here is an example of a pedigree. Now this says dominant pedigree up here. This represents juvenile glaucoma. It's a disease resulting from a dominant allele. Dominant means that you're going to see the allele expressed in every generation. Okay. Now, when we think just generally about pedigrees, these squares represent males. The circles represent females. If the square is filled in, that means that particular individual is expressing the allele, is expressing the trait. So here we have the trait is expressed, or an affected male. Here we see the trait not expressed, so this is an unaffected male. Circles represent females, so here we see in this female, the trait is not expressed, unaffected female. And here we see a female where the trait is expressed, affected female, okay? And so here you see an affected male crossed with, or mates with, an unaffected female. Now, this line here represents, and this little bar here represents their offspring, if they have any offspring. And in this case, this couple had 1, 2, 3 children, and they had 1 boy, 2 boys, and 1 girl. And they had 1 boy who was unaffected, and they had another boy who was affected, in other words, expressed juvenile glaucoma, and their girl also was affected. Okay, that's how that works. Now, this affected boy, right, joins together with this unaffected female, and they also have a child who is a girl, and she is affected, because remember, this is a dominant pedigree, so the trait, the allele, the gene, shows up phenotypically in every generation, right? So generation 1, generation 2, we see it twice here. In generation 3, we see it there. And here the affected girl mates with an unaffected male, and they have 2 girls, and both girls are affected. Okay, so that's how that works. And as you can see, juvenile glaucoma shows up in every generation. Okay, here's another one. This is a recessive pedigree for albinism. Albinism is a recessive allele, which means that it only gets expressed phenotypically if there are two copies of the gene for albinism. So while the trait may not appear in every generation, there are at least carriers in every generation. And what these little half circles and half squares, half colored, half clear, they represent carriers. So these are heterozygotes. So here you have, it says one of these persons is heterozygous. We don't know which one. We're kind of working our way back in this instance, but one of these is heterozygous. One would have to be heterozygous in order for the allele to be there to begin with, right? Okay, so this guy and this girl have 1, 2, 3 children. They have a girl here who's not affected. They have a girl here who is a carrier. She's heterozygote. And they have a boy here who is also a heterozygote, so he's a carrier. Now they're not expressing albinism. They're just carriers of the gene. This girl mates with this boy who is also a carrier. And they have 4 children. And because both parents are carriers, somebody is going to end up, some child is going to end up with albinism. And so they have a boy here who expresses albinism. They have a girl here who is unaffected. They have a girl here who expresses albinism, and they have a carrier, a girl who is a carrier. This carrier mates with this unaffected female. They also have 3 children. 1 boy, not a carrier, unaffected. 1 girl, unaffected, and 1 boy who is also a carrier. And so you see, while the trait may not appear in every generation, there are at least carriers in every generation. So here we don't see anybody affected with albinism. Nobody expresses albinism here, but you do have a carrier, right? In this generation, nobody expresses albinism, but you have 3 carriers. Well, you have 2 carriers from that couple in that generation. And this carrier mates with this carrier. Oh, I think I already did that one. Okay, and so then you have this carrier guy mates with this carrier girl, and they also have 3 children, a non-carrier boy, a non-carrier girl, and an affected boy, right? So the trait so albinism is recessive, so it can either show up in the generation, but if it doesn't show up in the generation, then there will at least be a carrier. So it'll either show up in the generation, but if it doesn't show up in the generation, there will at least be a carrier. Okay. So in a dominant pedigree, let's see if I can go back. Okay, can't go back. Okay, so in this video from the Khan Academy is a quick introduction to pedigrees. Okay, so worth your while to watch that. Now, sickle cell anemia or sickle cell disease has a heterozygote advantage, and what that means is that if you happen to be a heterozygote, in other words, a carrier of the sickle cell trait, then that protects you from malaria in those areas that are where malaria is expressed or where malaria is pronounced. Okay, so in tropical Africa, where malaria is common, so look, homozygous dominant, these are normal people, no sickle cell trait, they die of malaria. Homozygous recessive, all right, so if we're thinking about malaria, okay, capital M, lowercase m, if we look at it that way. So homozygous dominant would be big M, that's the normal person, no sickle cell trait, they die of malaria, there's no protection as a homozygous dominant. Homozygous recessive, that's little m, that means you have, if you have that genotype, then you express sickle cell anemia, and those people die of sickle cell anemia. But if you're a heterozygote, big M, little m, these are carriers, and they are free of both, they are free of malaria, right? They are protected against malaria. And these are just two little maps of areas of Africa where sickle cell trait is prominent. And you'll notice that the sickle cell trait is prominent mostly in those areas where malaria is prominent. And so the hypothesis there is in malaria-affected cells, the oxygen level is lowered enough to cause sickling, which kills the cell and destroys the parasite. So if you end up with the trait for sickle cells, that's the sickle-shaped red blood cells, then that protects you from malaria. Interesting that an otherwise problematic thing, sickle cell trait, can be a good thing in this situation. All right, so here's a quick check. Sickle cell is an autosomal recessive disease. Okay, both a man and a woman are heterozygous for sickle cell disease. Big S, little s, okay? Big M, little, whatever letter you want to use here. They're heterozygous, so big letter, little letter. What are the chances that any child born to them will have sickle cell trait? Not sickle cell anemia, but sickle cell trait. That means they're going to be heterozygous for the trait. We're going to let big A and little a represent the alleles. Big A dominant, little a recessive. I'll let you think about this. If you want to get out a piece of paper and a pencil or pen and work out a quick Punnett square, you can do that. So we have a heterozygous male mating with a heterozygous female. And you know what that Punnett square looks like. So here's heterozygous male or female, doesn't matter, and a heterozygous partner, see, big A, little a. And so the question is, what are the chances that any child born to them will have the sickle cell trait, will be heterozygous? All right, so if we do the cross, we have big A, 1, because remember, this is a simple monohybrid cross. So it's 1-to-1 genotype ratio, 3-to-1 phenotype ratio. Okay, so we're looking at sickle cell traits. So we're looking at the genotype ratio. So here's big A, that's homozygous dominant. Here's little a, that's homozygous recessive. And here is 2 big A, little a, 2 heterozygotes. These are your carriers. These are the people with sickle cell disease. These are the normal people. And so what are the chances? Well, 2 out of 4. Or 50%. Okay. Okay, so here's the deal, right? What is the genotype ratio? 1-to-1. That's 1 homozygous dominant, 2 heterozygous, 1 homozygous recessive. What is the phenotype ratio? That's 1, 2, 3 to 1. But the question is, what are the chances that any child will have the sickle cell trait? So this is, you don't look at it as if this couple has 5 children. You have to look at the chance per child. So with each individual pregnancy, what are the chances that that child will be a carrier? And that's 50%. Okay, the dihybrid cross now gets a little more complicated, but not that much more complicated. But here you're looking at 2 traits in a monohybrid cross. Mono means 1, so you're looking at 1 trait, 1 gene, 2 alleles but 1 gene. In a dihybrid cross, you're looking at 2 genes. Okay? So 2 separate traits here. So we're going to first of all start out with true breeding parents. That means true breeding dominants with true breeding recessives. Okay, and in this case we have the two traits are R and Y. So the dominant true breeder, right? So each parent has both genes, right? And each gene has 2 alleles. That's the way you have to look at this. So one gene is R, one gene is Y, but each gene has 2 alleles. And here we have true breeding dominant, that's big R, and big Y, big Y. And we're going to cross that with true breeding recessive, that's little r, little y, little y. So the F1 generation of a dihybrid cross is going to yield heterozygous or heterozygotes for both traits. So we have big R, big Y, little y, right? Only one dominant phenotype for each trait. And we're going to allow the F1, that's this generation here, the heterozygotes, to self-fertilize to produce the F2 generation. Okay, you get it? Right, the F1 generation are the result of this cross, and just like with the monohybrid cross, true breeders produce heterozygotes. But in this case we have 2 genes, 2 traits, so we're heterozygous for both. And then we're going to let these guys self-fertilize, or we're going to cross those. Okay, so then we're going to let the F1 plants self-fertilize. Remember, these are heterozygous for both traits. And that's going to yield the F2 generation and a set phenotypic ratio. In this case, it's 9:3:1. So remember, in your monohybrid cross, you ended up with a set phenotype ratio 3:1. In this case, we have 2 traits, and we have a set ratio of 9:3:1. That's 9 round yellow plants. Okay, this is seed shape and color. Okay, seed shape and seed color. So 9 plants yield round yellow seeds, 3 plants yield round green seeds, 3 plants yield wrinkled yellow seeds, and 1 plant of however many yields wrinkled green seeds. Okay, so here yellow is the dominant seed color, round is the dominant seed shape, and so we end up with only 1 out of the 16, 9, 12, 15, 16 plants is homozygous recessive, right? That's wrinkled, little r, and green, little y, little y. Let's look at it. Okay, so in a dihybrid cross, the alleles of each gene assort, remember, independent assortment, And the segregation of different allele pairs is independent as well, right? Independent alignment of different homologous chromosome pairs during metaphase 1 leads to independent segregation of different allele pairs. Okay, so we have the law of segregation and the law of independent assortment, and this is just reminding you, I showed you this before in chapter 11, I think, when we talked about the law of independent assortment, and that's why you end up with this 2 to the n number of chromosome combinations. Right, so in this case we have 3 genes, 1, 2, 3. So 2 to the third is 2 times 2, which is 4, times 2 is 8, 2 to the third power is 8. So you end up with 8 chromosome combinations. So keep that in mind as we look at the dihybrid cross. Because in the dihybrid cross we have 2 genes, right? We're looking at seed shape and seed color, and then when we cross those we end up with the heterozygote here, and we're going to cross those or let them self-fertilize, and we end up with 4 different combinations, 4 genotype combinations, right? So that's going to be 2 to the 4th power. So 2 times 2 is 4, times 2 is 8, times 2 is 16, and that's what you see over here. You see 16 combinations. So every time you add a gene, your Punnett square is going to get exponentially larger. So we went from the monohybrid with 4 squares to the dihybrid now with 16 squares, right? And it's going to go up from there every time you add a gene. And so what you see here is, okay, so big R, big Y is round yellow. Round is the dominant shape, yellow is the dominant color. And remember, heterozygotes are always going to express the dominant. So this is round yellow, round yellow, round yellow, regardless of what the genotype says here. If you have a dominant in there, it's going to be expressed round yellow, round yellow, round yellow. There's another round yellow, and there's a round yellow there. So that's 4, 5, 6, 7, 8, 9 round yellow plants. Then you have round green, and round green. That's 3 round green plants. Then you have round yellow. Wait. Then you have wrinkled yellow. So you have 3 wrinkled yellow plants, and then you have 1 recessive. So 1 wrinkled green right there in a 9:3:1 ratio. That's always going to play out. All right, always going to play out roughly 9:3:1 for a dihybrid cross. Good to remember that. So here's a quick check. So which of the following dihybrid crosses will yield the standard 9:3:1 phenotype ratio? Right? Look at them very closely. These are your choices. So which one of these will yield a 9:3:1 ratio? Remember, that's 2 heterozygotes crossed, or that's a heterozygote for both traits, dihybrid, self-fertilizing or crossed with itself. And remember when I said before, it does not matter which which allele, the dominant or the recessive allele, comes first. It does not matter, right? 3 times 2 is 6, 2 times 3 is 6, doesn't matter the order. You end up with the same answer. Okay, so let's look at them. Here is, we don't even care what the traits are at this point. Big C, big R, little r. That's clearly heterozygote, right? We're going to cross that with a big R and a little r and a big C and a little c. Now, here's the thing. It's the same same genotype, right? Again, it does not matter the order. So definitely that one is going to yield a 9:3:1 ratio. It's the same thing. You're crossing, heterozygous for both traits with a heterozygous for both traits. Here is big T, big B, big little b, crossed with a big T, little t, and a big B, little b. That should be obvious to you, right? Everything is in order there. Standard dihybrid cross, 9:3:1. Here is big O, big P, little p, crossed with a little p, big O. It does not matter, again, always does not matter the order, right? You can have the big P first, the little p first, it does not matter. You can have the O first or the P first, doesn't matter. You have to look at the genotype. What's going on in the genotype? 9:3:1 phenotype. Big A, little z, big Z, doesn't matter the order. Big Z, big A, little a, same thing. These are the same thing. Just jumble the letters, right? So all of these are going to display the 9:3:1 phenotype ratio. Be careful. Always look at what's going on genotypically. Don't take anything for granted there. Okay, a test cross is when you have an organism where you don't know what the genotype is. You see the phenotype, obviously, but you don't know what the genotype is. And in order to find out what the genotype, so it's an unknown genotype, in order to find out what the genotype is, you're going to cross it with something that you know. You know what the genotype is here. So you're going to it's used to determine the genotype of an individual with an unknown dominant phenotype. You're going to cross the individual with the unknown genotype with a homozygous recessive, right? Recessive for the trait, little p in this case. We don't know if this guy over here is big P or big P, little p. We don't know if it's homozygous or we don't know if it's heterozygous, right? Because we just see the phenotype. So in order to find out what the genotype is, cross it with something that you know what the genotype is, and that's going to be little p. And the phenotypic ratios among offspring are different depending on the genotype of the unknown parent. Okay. And so when you do that, then you're able to determine what the genotype is of that unknown. So the dominant phenotype, we get the purple flower. We don't know if it's big P, homozygous, or big P, little p, heterozygous. Right? So we cross it with a homozygous recessive. That's little p. We know what that genotype is. And so we have two situations here. So if you do the cross and you end up with all purple plants, that's what this represents here, then you know, right, the white flower is not being expressed. Then you know that the plant, you know that this plant is big P, little p, is a heterozygote. Versus if you cross the two, you don't know what the genotype here is, you just know it's heterozygous. You cross it with a recessive, little p. If you end up with some purple and some white, then you know that the genotype of this guy up here is big P, little p. All right. Let me say that again because I think I messed you up over here. So if you get all purples here and the recessive allele, the white flower, is hidden, then you know this guy, the unknown, is big P. And if you cross them and you get purple and white, then you know that the genotype of the unknown is big P, little p. Some genes have more than two alleles. The ABO blood type system in humans is a perfect example of that. There are 3 alleles in blood type, in human blood type. That's the A, the B, and the O. But remember that an individual can only have 2 alleles. You can't have 3 alleles, but you must have 2, which means that if you have blood type A, then your genotype is either big A or big AO. And if you have blood type B, then your genotype is either big B or big BO. You can tell now that O is recessive here, right? Because it's the B that gets expressed. Well, I shouldn't say, well, yeah, O is recessive. But if you have blood type AB, then your genotype is AB. So here's where you get into codominance, or in other words, these are both dominant alleles, but they're both expressed. All right? So that's where we get into the idea of codominance. You have both alleles, but they're both expressed, and that can happen. Now, if you have blood type O, then your genotype is OO, and O is not expressed, and we'll see that. We'll see why. So the ABO system demonstrates both alleles. So there are 3 alleles of the I gene, okay? It's I super A, I super B, and lowercase I, little I. We just use, just for clarification, ABO, but just remember it's this glycoprotein I that is what we're really talking about. So I super A, this is RBC, red blood cell, expresses the sugar galactosamine. I super B expresses the sugar galactose. This is the sugar on the surface of the red blood cell, on the surface of the membrane. And the lowercase I expresses no sugar at all on its surface. Okay? So codominance is the idea that in the ABO blood system, both alleles get expressed. So blood type A and blood type B are dominant to blood type O. So if you have AO, then A gets expressed. If you have BO, then use deodorant. If you have BO, the genotype, then B gets expressed. And the heterozygote, though, shows some aspect of phenotypes of both homozygotes. So if you have blood type AB, then both A and B get expressed. Okay, so in this video, it talks about multiple alleles. It's very simple, very kind of an elementary sort of illustration, a little animation, but it's simple visually, but it allows you hopefully to understand quite a profound concept here with regard to blood type and multiple alleles, and then Punnett squares. I encourage you to watch that. And so here's the deal, okay? We have the genotype. If the genotype is big A, right, you have 2 alleles, so you can have either big A or big AO, that's the small letter I, right, big B or big BO. You can have big A, big B, or you can have OO. Those are your genotypes. Phenotypically, big A or big AO yields A. You can express blood type A. Big B or big BO is blood type B. Big A, big B is blood type AB, and 2 copies of O yields blood type O. And the breakdown is here. As you can see, it's quite interesting, I think. So on the surface of the red blood cell, if you have blood type A like I do, on the surface of your red blood cell are these A antigens. These are these little spikes on the surface of your red blood cells. These are little sugar spikes. Right? Now, the deal is, though, if you have A antigens, then you have anti-B antibodies, or antibodies against blood type B, which means that if you have blood type A, you cannot receive blood type B because your antibodies will destroy the B blood cells because you have anti-B antibodies. So A antigens, anti-B antibodies. Can't mix those two types of blood. If you have blood type B, then you have the B antigens, these little sugar spikes on the surface of your red blood cells. But if you have the B antigens, then you have anti-blood type A antibodies, which means that if you have blood type B, you cannot receive blood type A in a transfusion, or you will die because your antibodies will destroy those red blood cells. That's the deal. Now, if you have blood type AB, you have both A and B antigens on the surface of your red blood cells, which means that you cannot have antibodies against any blood type, which means if you have blood type AB, you can receive blood from a type A person or blood from a type B person, which is why AB is referred to as the universal recipient. You can receive any type of blood because you do not have antigens to anything. Now, if you have blood type O, which is the rarest because this is 2 copies of O, right? OO, or little I, over here. You have no antigens on the surface of your red blood cells. No antigens, which means that while the rarest blood type, and by the way, you can only receive blood from another blood type O person because you have anti-A and anti-B antigens, but you have antibodies, but you have no antigens, which means that you can, if you have blood type O, you can transfuse your blood into any of these characters over here because there's nothing to reject. Okay, there's nothing to reject. So that would be universal donor. You can donate to anybody. Here you can receive from anybody. Here you can only receive from B or O, of course. Here you can only receive from A or O, of course. And here's a quick check. So if you have blood type O, then which antigens are present on the surface of your red blood cells? It says check all that apply. O and A. Now we're talking about antigens on the surface of your red blood cells. O and B. A, B, and O, or none. Blood type O. Remember, blood type O is a universal donor, which means that if you can donate to anybody, you do not have any antigens on your red blood cell surfaces at all. Okay? Preguntas, if you have any questions, let me know. And that'll be the end of this lecture. Thank you.